Good afternoon, everyone, and welcome to the Wake Forest Center for Artificial Intelligence Research, Artificial Intelligence in Action seminar series. I'm Metin Gürkan, Director of Center for Artificial Intelligence Research, CARE, and it's my pleasure to introduce today's speaker, Dr. Jingwen Yan. And Dr. Yan is an associate professor and director of bioinformatics at the Ludi School of Informatics, Computing and Engineering at Indiana University, Indianapolis. That's a difficult word for me. It has always been. Her research sits at the intersection of bioinformatics, computational modeling, and neuroscience, with a focus on integrating multi-omic data and neuroimaging to advance our understanding of neurodegenerative diseases like Alzheimer's. Her lab's work, funded by the NIH and NSF, contributes to national efforts such as AI for AD, Clear AD, and ADMC. In particular, her team is developing AI-driven approaches to identify interpretable biomarkers and model disease progression, and this work supports earlier diagnosis and therapeutic decision-making. Today, Dr. Yan will share recent advances from her lab using integrative omics to uncover molecular mechanisms and improve early detection strategies for Alzheimer's disease. Please feel free to submit your questions in the chat. We'll address them after the presentation. And if you're in the room, of course, you can ask your questions. And then with that, let's welcome Dr. Yan.
Thank you very much for the nice introduction. Do I stand or
however you feel comfortable?
Okay.
We are all right. Yes.
Okay, I will just sit here.
That's all perfectly
yes,
fine. Yes.
Easier to manipulate.
Of course.
To
Yes.
My laptop.
Yes.
Okay. Yeah. So thanks for the nice introduction. So today I'm going to go through summarize several of my projects. So and then you can ask a question at the end of the presentation. So, as Dr. Gürkan has introduced, my primary focus has been on Alzheimer's disease and recently extended to this normal aging. So the Alzheimer's disease is known as one of the most common type of dementia, so which accounts to around 60 to 80% of the brain dementia, like we already know. So it is known as a non-reversible and a progressive brain disorder that it's going to unfold over a kind of a decades of time, which will slowly destroy the memory and thinking skills. So inside this postmodern brain, it's not moving smoothly, but anyway. So in the postmodern brain, so people have found that two neuropathological hallmarks for the Alzheimer patients, like that within the neurons, you can see those kind of tangles. Those are known as neurofibrillary tangles, and these round plaques over here between the neurons are known as amyloid plaques. So if you look at this neuroimaging, like directly the MRI, so the Alzheimer patient on the right has a very much more severe brain atrophy. That means their brain shrinks, at a much faster rate. So the very starting point of our initial focus on AD is because like AD is highly heritable. So I think large-scale twin studies found that there is around like 70% of heritability, and some of them up to 80%. So based on that, they say like, oh, then why we should look at the DNAs. So they do this large-scale genome-wide association study to identify the genetic variations that are associated with this Alzheimer's disease. So particularly most of those, we call that GWAS, is focused on the one type of genetic variation, which we call the single nucleotide polymorphism. That is basically for this, you have for the two DNA copies from mother and father. So for most of these base pairs, we should be identical, but for some of these positions, single base pair that we can be different. So those positions are known as the SNPs. And once for the result of the GWAS, so you typically get a Manhattan plot, which look like this. So each dot is one SNP, and then their height indicating this p-value, the negative log 10 transform, the p-value indicates its significance in association with Alzheimer's disease. So, but then like with more and more GWAS has been done, there is a more like what the people ask, like what is the next step? So you identified those risk SNPs, then what are these connections between these genetic variations and this disease trace, like this imaging phenotypes we have observed from MRI? So how can we translate GWAS findings to some targetable functional mechanism in gene expression and protein expression? So that comes from our first research focus, which we have been doing for almost a decade. So in this one, we focus on these to identify or say to explore these molecular mechanism underlying those AD risk variants using from a multi-omics perspective. Like so, like from almost a decade ago, like so there is a huge increase of Alzheimer's disease data from not just the genetic variations, but also in the gene expression level and in the protein expression, even in the metabolite, as well as a wide range of neuroimaging phenotypes. So, and in addition to these omics data and imaging data, so there is also another rich resource of information is about how they interact with each other. There is a lot of bioinformatics curated database talking about this SNP is how they are associated with the gene expression, whether they're located inside the gene, or how the genes encode a protein. Protein can interact with each other. Proteins can catalyze these chemical reactions, the metabolic reactions. So we have all of those information out there. So, and what we have done several modeling work over that side, but the overall goal for this part of the project is like, instead of identifying one SNP, one gene, or say several proteins related to disease, we want to identify a pathway or say a disease module that actually can have some actual meaning. So we have developed multiple models along this line, and this is one of the most recent work on this. We developed a knowledge-primed neural network. We call that MOFNet, which is motivated by this original central dogma of molecular biology that DNA is transcribed into RNA and RNA is translated into the protein. So there is an information flow from the DNA to RNA to protein. So we build up, well, it's not a very traditional graph neural network. We will say this is a, if you think about this prior information flow, or say the prior knowledge graph as one network, then we use that network to wire this neural network over here. So this is a very simple neural network, but the first two layers, all these blue lines you have seen over here, are coming from the prior knowledge. How one SNP is connected to one gene, how this gene is connected to one protein. So in this case, it models the information flow from the DNA to RNA to protein. And especially if you think like for the first two layers, their connections are kind of one-to-one relationship in kind of mapping to this prior knowledge graph. So, and for the neural network, you can get a weight for these links. So when you get those weight, you can put the weight back to that prior knowledge graph that can prune your network. You can get a kind of disease-related modules rather than the single set of, say, isolated proteins or genes. So we use this data from two data sets, and because we have very limited data set for like having, because we require both these DNA, RNA, and a protein. So at that time, we did have very limited data. So we look at two cohorts from ROSMAP and Mount Sinai, so to see whether we can replicate our findings. So on the right side show are these prior functional interactions, or you can tell that's a prior knowledge graph we have about the relationships between these SNPs, genes, and the proteins, how we wire the first two layers in the neural network. So without deep into these more details, I'll quickly show you some of this performance. So we compared it with some of these other methods for just to classify the Alzheimer patients from these normal controls, and especially for this MOFNet, we have run 3 different versions. The green one you see here with the best performance is exactly what I have explained. The blue one, which is the second one over there, is like we shuffle this prior knowledge graph to see how that's going to affect the findings. And then for this light blue one, which is the third one, we remove those self-connections directly. So it's like I just integrate the information based on my knowledge graph. I do not use those measured omics data anymore. So, as expected, apparently, if you remove those omics measures, like in the gene and protein level, of course, the performance will drop significantly. But we also do see a significant increase so between the green and blue, that shows the value of that modeling the information flow, the extra value from modeling the information flow that gives you some boost in the performance overall across the different metric. So, and then we put a lot of time looking at our results. So when we map those weights back to this network and we prune this prior knowledge graph, and we get 3 major network components left. So this network is between the SNP, the genes, and the proteins. And the first thing we want to check is like for those SNP and the gene pairs we have identified over here, do they actually have some functional relationship? So when we select these SNP and genes, we select them simply based on their location. So the SNP is also located near the gene, but we do not know whether they have any functional relationship or not. So we go to the database called this EQTL. So EQTL database are basically they examine whether one SNP is associated with the expression of one gene. So, and that is a tissue specific information. So we go to the database and we found like for all the 53 SNPs we have identified, 50 of them are EQTLs in the frontal cortex tissue, which exactly where we collect our omics data. So that indicates some potential, like highly potential functional relationships between these SNP and gene pairs we've identified. But then we ask another question, is like this SNP, they are EQTLs, but they may not be the EQTLs of their connecting genes I identified here. So we further look at these SNP and gene pairs to see exactly whether this SNP has been associated with the expression of this gene in the frontal cortex tissue from another data set. So, and we found out of the 53, and we found 29 of them are actually can be validated. So, and since this data at that time, like we don't actually have single cell data, so we also look at what are these enriched cell types, because each cell type, they have their marker genes. So based on our genes and proteins identified in those 3 network components, we want to see which cell types are actually enriched. And we found like mostly enriched cells are basically astrocyte, microglia, and neurons, which are 3 primary kind of cell types inside the brain, which we find is very relevant. And most interestingly, finding from this paper is like one network component, which is a component 3 over here. When we search for the literatures, we find that all of these key members of genes and proteins, they are almost all of them are the members of this snare protein complex. So, and this snare protein complex is a key mediator of synaptic vesicle fusion, which means like from when the 2 neurons, they try to communicate with each other, they will form a structure called the synapse. And then how they communicate is like, so for this presynaptic neuron, they will have something called a vesicle. We call the synapse vesicle. So, and then these orange dots are those neurotransmitters. So, and then these SNAP protein complex actually will help bring this vesicle to the surface of that neuron. And then it will help these two membrane to fuse. So it will open up a pore, so those neurotransmitters will go out and be captured by these postsynapse. So we find this very interesting, and we further replicate this result actually in that Mount Sinai brain bank. Like, so this is a very small component, but we were able to replicate these findings, identify 3 out of these 4 proteins, 3 out of the 4 genes, and 4 out of the 6 SNPs in this Mount Sinai brain bank database. So that's kind of the first work we would like to talk about. So basically, MOFNet is a new model that we try to integrate this prior knowledge graph with the omics data to help generate some functional disease modules that can better provide a plausible, more plausible hypothesis for this causal molecular mechanism underlying AD. But we also see a lot of limitations. It's like because it requires all the subjects have all the omics data types, which kind of limit our sample size we can use. So, and we one of the current ongoing work is like we actually we revised the structure of this MOFNet to make it as we call that modularized MOFNet, so that within each modules, like it only requires one types of omics data. So that one types of omics data module can be pre-trained using the subjects with missing omics type. So that kind of you involve more samples into the training process, and we do see some improvement in this one. So, and also there's also some kind of concerns about whether you can identify like more connections. So that's not a kind of a limitation. I would say like the purpose of this one is trying to identify these the modules with known prior interactions. So it's not for new interaction discovery. It's only for for identifying relevant prior interactions. So, and for that study, what we assume, like from this, the result we have generated, there is one assumption. It's like one SNP is associated with one gene, but do they function by themselves? Do those GWAS SNPs function by themselves? So, say in the upstream of one gene, we previously assumed like there's one SNP over there. There could be multiple SNPs over there, and those SNPs, and in the genetics, so if they are the SNPs, if they are close to each other, physically close to each other, they typically follow a structure called the linkage disequilibrium block. And the SNPs within this LD block, they are typically highly correlated. And that kind of goes to this why you see this GWAS findings, they come as a clusters rather than one SNP at a time. So that's if it is more clear if you look at this result from the GWAS, you know, like it comes as one cluster. You see for this ApoE over here, one dot is one SNP. It comes, so it's not all of these SNPs, they're actually in separate positions. So you see them like all at the same position. It's because we just squeezed all of them together. When you spread them enough, you can see that these are just the nearby SNPs. So this LD block, what people do is like with these GWAS findings, like they think LD block is a redundant block. Okay, so they pick one SNP out of this whole cluster, and then typically they pick the lead SNP. Okay, so they find those with the most significant p-value, or they use this statistical method called the fine mapping to identify one of them to be the tag SNP. Then we ask the question that are these SNPs nearby really redundant? Like should we always just pick one? Are there any possible interactions within that? So, and then we did this project based on the recent advance, all about, not quite recent, on the deep genome annotation. So the deep genome annotation is kind of the project that, so if you have one segment of DNA sequence, like say one protein can bind it to that DNA sequence or may not bind. So, and also it can be, it has other, so this protein binding thing is called the transcription factor binding site. It can also be a DNA's hypersensitive sites. That means how likely it will be open up for some functions to happen, because most of the time the DNAs are in a compact kind of mode, like there's nothing will happen over there. So there's some work ongoing. It has been almost like quite established in the past kind of 10 years. They train a lot of kind of sequence-based kind of the big models, neural network models, that they have one sequence and then they predict whether one transcription factor will bind on a specific tissue. And then they will mutate. So you have one sequence, you put one SNP right in the middle, and then you can mutate that from minor to major allele. And then when you mutate, you can predict whether this transcription factor will still bind here or not. So if say like with major allele and minor allele, so the protein will bind when this sequence carries this major allele, but will not bind anymore when I mutate it to the minor allele. So that means this allele has a very kind of significant impact for this transcription factor binding. So, and this one gives you some more information about how it has been done, like about how the transcription factor binding to the center region and how that SNP is going to impact. But then the problem comes here, like for those GWAS regions. So if the center region is a GWAS region, and then you will have multiple SNPs located inside that, and how will that going to affect this final prediction? So we did some very, this is a side project that we did just for fun, but we find we have some very interesting findings. So we focus on only top 100 GWAS SNPs and also their neighbor SNPs around that 2000 base pair window. And then we use a trained model called the Expecto, which is from Princeton's group. So for this one, we first have that one DNA sequence from each SNP of interest, and that SNP is placed right in the middle. And then within that 2000 base pair window, you can have several other SNPs as well. But we just assume like they are the major allele. That means it's a reference allele. Everyone has the same. So we do not consider them. We only mutate this middle one. So this part we call that without the genomic context. That means we do not consider those neighboring alleles will have any impact. In the second step, we keep this center SNP as a minor allele, so that means it's mutated. And then we take turns, we make all these possible combinations, or we call that in silico mutagenesis on these neighboring SNPs, to see whether this predicted function of this sequence is going to change significantly compared to this without genomic context, or it will not change at all. So, of course, we have several results. I just want to highlight some of the most interesting part for this project. Like for those SNPs, like we find 4 SNPs with dominant allele effect. That means if you go back, look at these 2 groups of analysis, that means this sequence, like with only one mutation in the middle, has the same predicted function for compared to all of these groups with neighboring mutations. That means no matter how these neighboring alleles like mutate or not, so its predicted function won't change. So that we call that the dominant allele effect, which and then interesting, we find another 3 SNP that have significant effect of genomic context. That means when I mutate my neighboring alleles, the predicted function of this sequence has changed significantly. So these are the result for that 3 SNPs. So there are several rows. The first one is that the sequence carry both are mutated in both SNPs, and then the second row is like it's only mutated in one SNP, not the other. And the third one is like it's mutated on another one. And at the bottom line is the one like the sequence is without mutation in either of them. So you can clearly see for all 3 of these. Oh, it's not moving smooth. So for all 3 of them, so if neither of those pairs, like so for each pair, if neither of those are carrying the minor allele, so their log-off ratio is pretty it's almost neutral over here, it's 0. But then if you mutate one of them, you get some loss of function for this. For this specific one, it's this DNA's hypersensitive site in this astrocyte. But if the sequence carries these mutations in both SNPs, so the loss of function become even more worse over here. And we observed this for all 3 of them. And then we asked the question, so this is a very synthetic study. You know, the sequence is from the reference genome. The major and minor allele is from this kind of large cohort, from population. So I did not even look at the actual genotype data. So we go back to this adenine data set, and we use the actual genotype data to test for these interaction effect. So we were able to, so this one we did not have this data. So we don't have the genotype data for this SNP, so we couldn't validate this. For this pairs, we were able to validate their interaction effect in the adenine with, we get a very significant p-value. I forgot to put it over here. But for this one, we did not find any subjects in the adenine like with both mutations. So we couldn't do this interaction analysis. That also shows this limitation of this study, like which we're going to discuss here. So to summarize, this is a sequence-based functional annotation, like around these GWAS findings. And then we found like these genomic contexts actually have some impact on these SNP functions. And if we simply pick one out of this LD block for the downstream analysis, we possibly will lose some important information. Then we also found like these major and minor allele combinations we test is synthetic. So, and those combinations, some of them, they may not exist in the real population, just like when we try to validate that in the adenine. So for some of them, so for one of them, we couldn't even find those subject with those. So, and for this part of the work, I mean, there is some like recent like long read sequencing that possibly could, if the data is available, that could be possible to help reveal some real haplotype or these real allele combinations in the sequence that we can leverage for the further investigation. So that is kind of the primary focus of my work during my tenure track. And then later, we have been shifted our focus, expanded our focus a bit to this another topic called the Alzheimer's progression profiling. The goal of this direction is to detect when and how multi-omic molecules and imaging phenotypes change along the decades of progression process, and which hopefully can help with this early detection of Alzheimer's disease. So this has been a topic like in the field for quite some time, and especially a lot of work has been looking at this. So say a few biomarkers of Alzheimer's disease and which one comes first and which one comes later. But most of these, they are, I would say, they're based on the statistical method. And then, and they only look at a few biomarkers. Okay, so, and currently the framework that has been used for these Alzheimer classification and quantify their progression is a framework called this ATNVI, which means A means amyloid, tau, T is for tau, and N is for neurodegeneration mostly. So amyloid and tau are mostly captured by PET. Neurodegeneration are mostly captured by these MRIs. And then they also measure the vascular and inflammation biomarkers. But I mean, these are mostly ranked by their prioritized. So the amyloid has been one of the driving factors to determine whether a patient will be diagnosed as AD or not. So, but this current framework, they are based on the binary classification for each of these group. So basically they're going to discuss whether this is amyloid positive or negative case, or tau positive or negative. So, and you just look at the combinations. So if you only have ATN, that means there is only 8 possible combinations out there. So, and we see so this is still discrete, or say kind of the stages. Why can't we just do a continuous quantification of this AD progression so that we can fully capture that the whole process, which is decades long? And another kind of the advantage of this continuous quantification is that for the early stage, if you use those, say, amyloid positive or negative, or the diagnosis normal control, EMCI, late MCI, and Alzheimer disease, so those early stage are treated without any differentiation. They're all amyloid negative, but there must be something happening within that. If you do a continuous quantification, you can see within that short period of the preclinical stage, what is possibly what is happening over there. So, and based on that, so we first started with some kind of existing work. So we didn't do our own models at this point for this one. And then we say we borrowed some ideas from the single cell analysis recently. So for the single cell analysis, there is a type of kind of approach called the pseudo-time analysis, or known as a trajectory analysis. So what it does is if you look at the right side, so it tries to capture this developmental trajectory for the cell differentiation. Think about originally, we have these stem cells, and the stem cells get to differentiate, go through some intermediate cell states, and then differentiate into mature neurons, mature astrocyte, so or red blood cells, and then but this process, it's there. However, when you try to capture the data, you will not be able to. You capture a data, you get a set of cells. So you get a set of cells that you get a little bit of everything, but it's a set. You don't know which are stem cells, which are the mature neurons. So the pseudo-time analysis actually aims to recapture the pseudo-time, or the temporal information, from the snapshot data. So, and then we say if they can do this, why can't we do this for this patient progression? The patient progression needs a similar kind of this longitudinal this process. If they can use this from the snapshot data to recapture that temporal information, or we call that pseudo-temporal information, we should be able to use this for this disease progression as well. And then, so we say okay, we will just start from this, and we look at the multimodal imaging from amyloid, tau, and neurodegeneration to see which one can actually better capture that AD progression, and whether the findings is aligned with what the current kind of observations. So the work, we tried actually a lot of approaches, but then we ended up with a fate, which the basic steps is you have your original data matrix. So say in the cell, they have cell by genes, but in our case, we have these subject by brain regions. And then we build up a affinity matrix, or say similarity matrix, and then you do this diffusion process. So why the diffusion process? Because if you only do this, so it's basically a graph embedding problem, you do this graph embedding from the similarity graph, a matrix, or similarity a graph. But if you do that without any diffusion process, you won't get that nice progression curve. So it's not as compact. So the diffusion process helps to generate a trajectory that is compact and smooth, so it can be better viewed as a progression process. And we use the ADNI data. We look at the MRI PET, amyloid PET, tau PET, as well as a small subset of them, like with all three imaging modalities. So this is a final trajectory actually look like. So so on the left side, for this cognitive normal controls, so they're mostly on the left side, and for these AD, which are the purple dots, they're mostly on the right side. So different column are for different imaging modalities. So the first thing we want to examine is like, you need to at least have some metric to evaluate your trajectory, because this is purely kind of, unsupervised, and then it's purely a discovery analysis. So one thing to validate is like, the trajectory need to have, say, these cognitive normals need to have a lower pseudo time, on the one side, and then these Alzheimer patients should be on the other end. So because we want this overall curve from left to right mimic the progression stages of this the Alzheimer disease. So then for each of them, for each of the imaging modality, we look at this pseudo time. So the pseudo time, if that means the relative location on this curve, if they're on the left side, that would be the pseudo time is 0. On the right side, the pseudo time is 1. And then that can also be used as kind of, the staging of this based on this specific modality. So, and then we can see that for these cognitive normals and then these early mild cognitive impairment patients, so their pseudo time are mostly at the lowest for almost all of them. And then the Alzheimer disease patient has the highest pseudo time distribution, on which means that they're mostly on this end of this principal curve. So that kind of, to some extent, like validated, like, so it captures at least the progressiveness of diagnosis groups. So, and then we found something very interesting from this multimodal pseudo time. So in I think several years ago, there is a hot topic about the amyloid clock. So the basic things like the amyloid accumulate not in a constant rate. It starts very slowly, but then once it reaches a certain point, it's going to start to accelerate. So, and then we see this is a pseudo time, and that to kind of, to some extent, represent the disease stages. So we want to see if we plot the pseudo time against these overall amyloid deposition, can we capture that amyloid clock or the turning point, the acceleration point? So we look at the pseudo time that captured by the amyloid data itself. So we did not see anything, but if you look at the multimodal imaging-based pseudo time, we can see there is a clear turning point over here, which is right around this 1.2, which is exactly what they have previously observed in another study. So that kind of thing, this finding, actually get us really excited, shows oh, okay, so this pseudo time can, it does work to some extent. And then we also look at another thing at the end over here is so, okay, this turning point is around between 0.4 to 0.6. So we look at those Alzheimer patient with this risk allele. So one of the top risk allele for Alzheimer is called ApoE4. So if a patient is amyloid E4 positive, that means they carry that allele, then they have a higher risk of developing AD. So we look at that. So does those patients carry this ApoE4 allele? They reach this turning point faster, or say at a younger age, than those who do not have that. So, and then we look at that. So this is turning point data. So this blue or green one, so that indicates the age for those E4 positive participants. So what is the age they reach this turning point? So it is much younger than those ones who is amyloid E4 negative. And if you look at this later stage, so say this later stage over here, we observed a similar pattern. So these E4 positive patients, they reach the turning point at a much younger age, and then they reach the most severe AD stage also at a much younger age. That kind of confirms this impact of this E4 allele. So, and then we later also look at, say, along this pseudo-temporal line, like what are these order, or say the temporal order of those imaging phenotypes? Do they actually align with the previous findings? You know, those has been done with these statistical models previously. So, and there have been a lot of work like previously, those are the hypothetical model, like orders has been raised and clinically kind of observed for some of them. So they found like amyloid is started to accumulate first, and then they have these tau comes after that, the neurodegeneration and the tau, and especially these MRI and tau PET, they are very quite close when they have these abnormal changes observed. So then we plot those different multimodal images. So we look at their normalized image measure, how that change along this multimodal pseudo time. And then so for on the left side is for the multimodality. So the MRI does not have a significant decline until the late stage, which is a red line over here. The amyloid is the green line that actually start to show acceleration. It start to accumulate pretty early, but then have acceleration around this stage over here. And tau and these MRI have a very similar kind of the changing point over here, while the MRI shows the brain atrophy, which is decline, and then the tau shows the tau deposition, which increase. So their timeline are very, very close, which is quite aligned with the previous findings. And then next we look at these imaging phenotypes within this modality, particularly for tau. So the tau, like they have some, so for Alzheimer patients, they have another kind of staging score called the BRAC. So the BRAC basically is based on examining the tau deposition, like, or say tau spreading. So they assume the tau spread follow a certain path. So if they see the tau so only in this entorhinal cortex, that's a BRAC stage 1. If they see this, it's spread into the hippocampus, then that BRAC stage 2. So based on that, they define these different regions of corresponding to the BRAC 1 stage, BRAC 3 stage. I actually skipped 2 because the hippocampus data has been contaminated, so we exclude that. So, but then we separate these brain regions into different stages. That means the tau deposition in these brain regions won't occur until the late stage, but this the tau deposition in this BRAC1 region will occur very early. So when you look at these lines, you can clearly see that BRAC1 stage, like this line goes up very much much earlier, but then this BRAC3, 4, and 5, 6 like comes way later over there. So that's kind of from another kind of point of view, like confirm this potential of the pseudo-time approach to continuously quantifying this disease progression process. And then later we find another thing is like there is a lot of work talking about that individuals with substantial amyloid burden that they can still remain cognitive normal. So that means the image itself sometimes is not enough. So, and then also there is another paper talking about, you know, like they think like the rate of cognitive decline also depends on the genetic background, you know, so that kind of contributes to this heterogeneity in this cognitive decline, even with similar amyloid burden. So, and then we see like, okay, why don't we, when we quantify this progression, why don't we integrate this amyloid imaging together with our GWAS findings? So we still look at the ADNI data, and then in this one, we only focus on the amyloid PET. And for these GWAS findings, we pick this top 27 AD risk SNPs that has been widely used for the polygenic risk score. So, and then this gives you a rough idea how we integrate them. So we didn't directly concatenate them together. Instead, we build up the similarity network for each of them. And then we run this similarity network fusion. So we ultimately get a fused similarity network that will go through this diffusion process and graph embedding to get the final progression trajectory over here. So this is the result we get actually. So the curve is not as smooth as the previous one, which likely due to the sample size. But we still find out like these the progressiveness between these diagnosis groups has been well captured over here when we examine their pseudo-time distribution across diagnosis groups. So we did not use this as a staging score, unlike the previous one, because the genetics does not change. So it's not a proper idea to say to interpret this as a staging score. So what we do is like we want to use this fused kind of pseudo-time as a potential risk score. So to stratify the patients, see like how likely they are going, how fast their cognition will decline, and how likely they're going to, or say how fast they're going to progress to Alzheimer's disease. So we first look at their correlation with longitudinal cognitive performance. So we cut for this, so this is only on MCI, as I said, like, so this project is trying to, for this early risk assessment. So we focus only on this MCI subject that have some memory problem, but hasn't been diagnosed as AD yet. So, and then we separate them into two groups based on the pseudo-time, like it's below 0.5 or above 0.5. And we also, they can also be separated based on their diagnosis. They are early mild cognitive impairment or the late mild cognitive impairment. They can also be separated by this E4 status, that risk allele, whether they carry it or do not carry it. They can also be separated by the amyloid positive or negative that has been used in the ATM framework. So we want to see like which of them can actually have a better stratification power to differentiate two groups with different cognitive decline rate. So for all of these four categories, so these we find this significant result for two cognitive measures, that is ADAS 13 scores and this composite admin memory score, which is a composite memory score they have come after. So we found this pseudo-time actually works pretty well, also similarly as this amyloid positivity, but not for the diagnosis and APOE, this risk allele carrier. So none of them actually can differentiate these two groups with faster or slower cognitive decline. And we get the most significant p-value over here for the pseudo-time on this composite memory score. And then we also want to see like how likely they're going to, or how fast they're going to progress to this Alzheimer's disease, because for the mild cognitive impairment, like their concern is like whether I'm going to convert or not. So, and then we look at this traditional compare this pseudo-time with other kind of criteria, diagnosis, is allele carrier and amyloid positivity. So actually like all of them can stratify the MCIs, but these pseudo-time has the highest, has the lowest p-value and the highest hazard ratio detected. Oh, almost time? 5 minutes? Okay. Okay, and then the next question is like, if those patients, so say for these subgroups identified by the diagnosis, so say they are eMCI, or say for those groups like they are already amyloid positive, can the pseudo-time further stratify them into even smaller groups? So, and then that's how we did this kind of differential progression within these MCI subgroups using the pseudo-time to separate them into two groups. And we found that the pseudo-time can further separate these subgroups into these high risk and low risk in all of them, especially for this amyloid positive. I think we mentioned earlier that for the amyloid positive, they can still remain cognitive normal. So so this score can be used to further stratify those amyloid positive to lower risk and higher risk ones. So it will be very valuable for this. I mean, when they try to for the clinical trials, when they try to identify the highly risky groups. So, and we replicate this finding on the validation group, like we purposely set aside some subjects aside because this is unsupervised like discovery analysis. So it's kind of tricky, like it's not a pre-trained model, you can put the tested data directly on that. So we did some kind of tricks, like we add one validation subjects point to this training one at a time, which, I mean, of course, that's a limitation. You need to retrain everything from scratch. But because you only added one at a time, so overall, it does not change your previous result. But at the same time, you get a pseudo time for this test subject. So we were able to replicate this stratification power of pseudo time for both cognitive decline and this the progression risk, but not for any of the others criteria, the diagnosis group, ApoE, E4 carriers, and amyloid positivity. So we weren't able to replicate for any of those. So to summarize, these two studies in the second research focus demonstrate the potential of the pseudo time for AD progression. But as you can see, like both of them are discovery analysis with limited capability to generalize. So, and also they leverage only cross-sectional data. And so we do have many of the modalities have short-term progression patterns that we could be leveraged. So, and our current work is trying to build some new supervised training methods that can be directly applied to the new patient, and also at the same time to leverage the longitudinal data. So due to the time constraint, I'm going to stop here and I will stop this. I have 3 extra slides for this modeling side. I will skip that, and I would like to acknowledge my collaborators, students, as well as the funding sources.
Fascinating work. I was just wondering where I sit in my amyloid clock. It's really good. So the floor is open for questions. Does anybody have any questions? I have several, but just wanna. Do we have any questions in the chat? So if you want to ask your questions for those on the online, please put them in the chat or raise your hand. Not the physical hand, a virtual hand. So we will see you. You touched upon this a little bit. Just part of the your talk. You talked about the variations, and some people do not have any signs of the amyloid development. They may show amyloid development, but their cognitive functions may be intact. Yeah. And I think I remember a study with French nuns. And then they are just, when French nuns donated their brains to science, they cut them up, and then they saw lots of amyloid development, but their cognitive function was intact, right? And I said, well, there is no way I can be French or I can be a nun. Just I'm not it's not going to help me. But I wonder if it will be helpful to look at, I don't know if their data is available, and how do these people's, data would fit into this amyloid clock. And then the other thing that came to my mind is just, there are now, I think, FDA-approved drugs that can reduce amyloid development. And then when you take their data and then put it in your, pseudo-time analysis, do you think that you will be reversing the clock and then you can see the shift coming back? So these are speculative questions, but I just wanted to see what you think.
Yes, I think that's a very interesting question. So I mean, for this progression part of the work, like what I presented today, they're mostly like what I used as a preliminary data in my proposal. The actual work we're doing right now is developing new models to track that progression, and also that going to be applied to the new patients.
Right.
When they come in, you can project where they are.
Right.
Yeah, and then talking about whether that can be reversed. So that one I don't know. So this whole work, like I think that when we try to say shift from this discovery analysis to this deep learning model based analysis, is coming from these single cell foundation models.
Mm-hmm.
Like so they have a word called in silico perturbation. So basically, if what we're trying to do now is you can learn a kind of progression trajectory, like using the deep learning models, and then you can knock out some. So say if it's a gene based,
yeah.
So then you can knock out some of those to see whether it will go backwards. So that's kind of for Alzheimer's disease. I really don't know.
Yeah.
But yesterday I had a conversation with Dr. Harrington, so he's doing very similar work, and we have a similar interest. But for the cardiovascular disease, it is possible.
It is possible.
It is possible. But the thing is, the tricky thing is, you can, because it's in silico perturbation. That means you knock out only one.
Yeah.
So, but the whole system is very complicated. They interact with each other. I mean, those foundation models, they have mentioned that they, to some extent, they model this context information between the gene interaction. They kind of account for that kind of interaction context information, but how much that has been counted, so we don't know.
Okay.
Yeah, so I think that's kind of one of the most exciting points when I go along this direction, but we certainly don't know whether that's possible. And especially for the Alzheimer's disease, it depends on the data you have.
Right.
Yeah, and those drugs, I don't know. It's the drugs they approved, but I just read a paper from, I think, the Lilly one, the A4 data, the amyloid aging thing, they have published the paper says the Lilly one does not have any effect.
Yeah.
So I think it's very controversial for
Yeah.
both of them, for both drugs that has been FDA approved. I think there are still some debate and controversial whether it actually works. But it's definitely not there yet.
No, it will be interesting if you can find their data to apply it, and if you see that you are taking the clock back. I think it will be really, really interesting to see that.
Yeah, we are currently collecting the A4 data.
Okay.
So yeah, we just got it downloaded last month. So we were trying to see whether we can get some of those information, because this clinical trial, the treatment information for Alzheimer's is not that much.
Okay.
Because most of them, they failed, the clinical trials fails very early, in a very early stage.
Yeah, that's interesting. We have a couple of minutes left. Do we have any? I have other questions too, but do we have any?
Dr. Ma raised his hand.
Oh, okay. Yes, Da. Please go ahead. Yes. Turn your microphone on. Ask your question, please.
Yeah, thank you for the fascinating talks. Just a couple of minutes. I have one short question. So I think the pseudo times and the staging information is really clinically sort of relevant, and you mentioned about the potential for clinical translations. I was wondering whether you need multimodal data, both imaging and genomic and multi-omic data for that, and especially in early stage, would it be when it's hard to collect a lot of the multimodalities? How can we handle the sort of missing modalities, like what you did in your first stage, where you train the foundational models in each modalities and then combined? And maybe a related question is that how that is affected by the age. How can we control that in interpretation?
Yeah, I think for the first question you're asking about the second project, right? The second focus on the progression side. Yeah, so I think it has it has the same problem with the first one, is about you're missing data. So it happens. Like I think we're currently, I would say for the multimodal work we have done so far, like for the second part, it still requires that all the subjects has all of the omics data. But yeah, but yes, I think like the next step is like if we want to look at this multimodal progression, and then to address that missing data problem will also be very important, because like you do not want to narrow. So say like that these amyloid clock work. So that one you can clearly see their progression trajectory does not look as nice as others because the sample size is so small when you overlap across these modalities.
Exactly.
So the second question is about the age. Can you repeat that question?
Right. The second is, I think
the second question needs to be taken offline. I apologize. We are over our allocated time. So I just want to thank everybody who attended the talk, and thank you, Jingwen, for the fascinating talk. We have a lot of questions, but we'll follow up with you. We'll be watching that clock. That is another clock for us to watch. And then, let's thank our speaker one more time.